Hey guys, so we are going to start our biotechnology unit. I'm going to do 4 videos. So on our schedule that we're giving you, there's outlined what slides you're supposed to be reviewing each day. So I'm going to do 4 videos to go along with each of those 4 days. So this is the first one. So let's start biotechnology. This is the manipulation of living organisms or their components to produce useful products. So this is super relevant to us right now. The most relevant thing that I can think of is that this is also used to create vaccinations, and I don't even have to say why that's relevant to us, but they are working right now to create a vaccination for the coronavirus, which would obviously be super helpful for everything that's going on. It's also used for pest-resistant crops, new bacterial strains, and novel pharmaceuticals. And so the engineering of organisms for useful purposes is that short definition. So yeah, it can be used for so many different things. It's crazy and it's super helpful, and a lot of the stuff that we use today and don't even think about being biotechnology so it's kind of a cool unit. So topics in biotechnology that we're going to cover: DNA manipulation, DNA copying, cloning, genetically modified organisms. I'm sure you've heard of GMOs, so we'll talk about those later. Genetic screening, and gene therapy. All right, manipulating DNA. Scientists use several techniques to manipulate DNA. Chemicals, computers, and bacteria are used to work with DNA. And we'll talk about how they're doing this and what exactly they're using later. Scientists use these tools in genetic research and biotechnology. So basically, the whole deal is that they're manipulating this DNA to do things other than what it naturally would. So restriction enzymes, pretty cool. They're like little molecular scissors. So they're going to cut our DNA, or cut the DNA that these scientists are working with at specific spots. They're going to look for bases in order, and when they find the bases in order that they are supposed to cut at, they'll cut the DNA into fragments. So it comes from various types of bacteria, allow scientists to more easily study and manipulate genes, and they're going to cut our DNA at a specific nucleotide sequence called a restriction site. So restriction enzymes cut at restriction sites. Should be easy to remember. Okay, so different restriction enzymes cut DNA in different ways. Each enzyme has a different restriction site. So this enzyme may cut at AACT, and the other enzyme may cut at GGCC. So they just have different sets that they're looking for, and that's where they'll cut. So you can see here, restriction enzyme 1 would end up cutting the DNA into 4 fragments, where restriction enzyme 2 would cut it into 7 fragments. This doesn't mean that the restriction enzyme is set to cut into 2 fragments or 4 fragments or 7 fragments. It's looking for those bases in order, those sequences, and that's where it's going to cut. It's not really about how many it's cut into. Some restriction enzymes cut straight across and will leave blunt ends, which is going to be like it cuts straight down, and there's going to be the same amount of nucleotide bases on one side as on the bottom side. And then some enzymes are making staggered cuts and leave sticky ends. Sticky ends help ensure that human DNA fragment is inserted into the plasma in the right direction. Talk about that a little later. But you can see here, these right here, these sticky ends, they're off balance. So there's more on the top as there are on the bottom, or vice versa. And so you can see over here, it says a restriction enzyme cuts DNA when it finds a restriction site. And its restriction site is TCGA on the top and AGCT on the bottom. And so if one of those letters was changed, it may not cut there because it's looking for that very specific sequence. And the last thing we'll talk about today, gel electrophoresis. There really is a good video by Amoeba Sisters on gel electrophoresis. I even miss hearing you guys whine about Amoeba Sisters. This is super sad. Gel electrophoresis is used to separate DNA fragments by size. So DNA sample is cut with a restriction enzyme. So that's the first step, is they're going to use one of those restriction enzymes. The electrical current pulls DNA fragments through a gel. So we're going to start with this gel. The DNA is placed at one end after it's been cut by this restriction enzyme. Then they're going to turn on this electrical current, and it's going to pull the DNA through the gel. As it pulls, the smaller fragments are going to move fast and the larger fragments are going to move slower. So then after looking at the gel, you can tell, oh, this is the longer one because it's closer to where we started, and this is the shorter fragment because it's farther away from where we started. The fragments are going to appear as bands on the gel, and bands can then be measured and compared. And you can see it up there. It's what it kind of looks like after it's been pulled, after the electrical current's been turned on and the DNA fragments have been pulled through. A restriction map shows the length of DNA fragments on the gel between restriction sites. It only indicates DNA size. Sorry, tiny little dogs just came out. Not the sequence. It's useful in genetic engineering, used to study mutations, used to help determine paternity, which means who's the father, and then used in forensics. So if I had two suspects and I had DNA from a crime scene and I cut all of those DNAs, like strands, with the same restriction enzyme, and I did gel electrophoresis, the bands would be pulled the same if the suspect matched the crime scene DNA. I kind of said that really weird, so I hope that made sense. If not, I'll rephrase. These dogs are really cute. They're just really annoying. Alright, so you can see right there, that's what it looks like if we'd started. They're on the top and the DNA fragments are being pulled down. The direction of travel is down. And so you can see that that orange one is the smallest. It traveled the farthest, and the green one is the longest, and it traveled the least amount. We'll talk about PCR later. So that is all for now. Let me know if anything didn't make sense or if you need me to re-explain. Miss you guys. Have a great week 2 in quarantine. Hopefully we'll be back at school soon.